And now it's my great pleasure to introduce Dr. Mirella Dapretto. Dr. Dapretto is a professor here in psychiatry and biobehavioral sciences, and she heads up several of the projects, both the infant imaging and the longitudinal imaging projects in autism at UCLA. Dr. Dapretto has been with us for many years. She's a UCLA undergraduate, graduate, postdoc, and faculty. So definitely one of our own, and a brilliant experimentalist. She'll be speaking to you about functional brain imaging research in autism, particularly focusing on sensory over-responsivity. So please welcome Dr. Mirella Dapretto.
Good morning, and thank you, Susan, for that very kind introduction. I hope it didn't raise too high expectations for my talk. It's always hard to follow Dan. I'm the least morning person, and yet I'm speaking at 9 a.m., so you'll forgive me if I make sometimes not much sense. Please do raise your hands and ask any clarification question as I'm going along. On the bright side, I may not speak as fast as I typically do, because half of my brain is not synapsing. So we'll see how that goes. Let me start first with the acknowledgement, just in case we run out of time. In particular, I want to thank among my collaborators, Dr. Susan Buchheimer, who was my mentor, and we've been doing research on autism for the past 20-some years now. So. And I also want to acknowledge our funding agency, particularly a grant from the Salience Foundation, who is now funding our work on sensory over-responsivity, a great list of fantastic trainees, and there are two missing among those because they deserve their own acknowledgement, since I'll be talking about their work at great length indeed. Shula Green here, who's a postdoc and is in the audience there, should have been the one giving this talk, because a lot of it's really her work, in both as she was completing her Ph.D. here at UCLA, and the work that she's conducting now as a postdoc. And Tani is actually a third-year graduate student in the developmental area, and she is she's like basically taking the lead in our studies in infants at high risk for autism, and I'm going to be touching upon those as well. Okay, after this due acknowledgement, a brief outline what I'm going to try and cover. We're going to talk briefly about what sensory over-responsivity is all about, of course, as it relates to autism, but I need to point out that it's also evident in a number of other developmental disorders. I want to talk mainly about the mechanism that underlies sensory over-responsivity, and we're going to be looking at both studies that Shula conducted that shows both altered brain responsivity to sensory stimulation, so altered brain activity, as well as altered brain connectivity. Again, I'll also talk a little bit about our work in that area in the infants that we're seeing, and then I'll finish up with just a hint of what the field is, where the field may be headed next. Now, looking back at some of the slides at the talk that I gave 2 years ago at the symposium, Susan and I alternate, I realized that I had put in my future direction slides 2 years ago a couple of papers, one of which I'll tell you in a second about Shula, that had just began to address the neural correlates of sensory over-responsivity. Now, it's interesting because while sensory abnormalities have been described since the early first description of autism, it's only recently that they've become into sharp focus, likely due to the fact that the DSM-5 now actually highlights sensory abnormalities as one of their diagnostic criteria. And indeed, so much has been happening in the past few years that I would not be able to cover the number of studies that have been published since then. But what I wanted to highlight that is this is really like taking it's affecting not just the science that we conduct, but also it's bringing awareness of the issues that many children and their families are facing when children have such problems. And I just came across this very interesting ad a few days ago that shows that the AMC theaters are actually now having the sensory-friendly films, particularly geared toward families that have children with autism or with other disorders that have this issue. So I think something good is definitely coming from this renewed attention to this aspect of the core deficits we see in autism. Okay, so sensory over-responsivity, it's extremely common in autism. I mean, rates range between 56 and 70%, but actually if you consider also children that may have hyper-responsivity, that is decreased responses to stimulation, can be even higher than that. And that compares to much lower rate in the in neurotypical children. But as I said, you see this similar sort of deficit in other developmental disorders, including ADHD, Fragile X, fetal alcohol syndromes, and so on. So I think it's important to, again, acknowledge that there is this common path across different developmental disorder, which can actually inform our understanding and hopefully the treatment that eventually may be developed. Okay, so its over-responsivity, it's characterized by negative reactions or avoidance of sensory stimulation. And this can range from loud noises to very intense visual stimuli or like flashing lights, etc. And also to particularly to touch. And like children may really have issues wearing certain type of clothes, etc. It's associated with lower social and adaptive skills in children with autism. It is also associated with more internalizing problems, especially anxiety. And overall, it's taking a toll on not only on the children, on their families. Now, this is a condition that it's basically, for the most part, assessed. I mean, this deficit are assessed by parental report and from overt behavior. And that is one of the issues that I'll come back to later as to areas where the field needs to do more work. So in Schuler's first study, which was published a couple of years ago, she basically pioneered, I believe this was the first study that looked at the neural correlates of sensory responsivity, and she presented in the scanner during a functional MRI task these two sets of stimuli, and let me see if it works. So it wasn't particularly disturbing, but you have to imagine that you are wearing some goggles and that this is really like in your face and occupying the entire visual field. And then in other conditions, they would hear just this much louder, didn't want to make it too loud, like a very strong white noise. And then there was also a condition where the two were presented together. And what she found was that there was indeed greater responsivity, greater activity in children with ASD as compared to children with typically developing. So the TD stands shorthand for typically developing. So you see similar patterns of activity in both visual areas, of course, and auditory cortices. And some of these regions here are, you may be aware or not, it's part of the amygdala complex, and it's a region that it's involved in arousal, in detecting salience information, in emotional responses. So here are the regions where the children with autism show significantly greater activity, and what she did later was to examine how sensory over-responsivity modulated responsivity in the brain. So basically here we're just comparing how the brain responds when presented with a different type of stimuli. In this set of analysis, instead, we take the children's scores based on a variety of measures I'll describe in a second, and we looked at whether those scores modulated activity in the brain. And you can see that there were a very strong relationship in both the typically developing children and the kids with ASD, whereby the more the parental reports of social over-responsivity, the greater the responses in this amygdala regions that are important for, again, detecting what is Salience and for showing the negative, in this case, likely negative emotional responses to the stimuli. Okay, but Shula did not stop there. She immediately then thought about the next step, and this is a paper that came out already last summer. And in this paper, she extended the prior work considerably by first looking at a different type of sensory stimulation that it's known to that it's particularly discriminating between children who do have SOR versus children who do not. So in one condition, she again had auditory representation. This time it was not auditory, white noise, but rather a range of environmental stimuli like loud honking, etc. And then, I mean, this may not really give justice to what we did, but while the children were being scanned, we coordinated. We had one research assistant inside the scanner that was rubbing the inside of the arms of the kids with what was a wool cloth that was kind of abrasive and irritating. And then we also had a condition where, again, we paired the stimulation, the tactile stimulation, as well as to the auditory stimulation. And the behavioral measure that I alluded earlier that she collected, we used the short sensory profile and the sensory over-responsivity inventory, but we focused on a composite score whereby we added the standardized scores on some subscales of both measures that pertain to auditory filtering and visual sensitivity, auditory sensitivity, and tactile sensitivity. And we also use the screen for child anxiety-related emotional disorders. I mentioned before, SOR is highly associated with anxiety, and we wanted to make sure that we could dissociate the effect in the brains of the two of the two mechanisms. So, not surprisingly, in this study we had a larger sample, so we actually could tease apart in the children with autism. We had a group that had elevated scores on the SRR composite scores. These are the kids whose composite score was in the top 25%, and a group of kids with ASD who did not show any strong SO problems. Same here for the typically developing kids. Now, again, the scores here show that there was much more anxiety in the children that do have sensory over-responsivity. So here are the findings, and you can again see in the typically developing kids and the children with autism, there is some similarity, of course, in the pattern of responsivity. I'm showing you here the results for the joint condition where they were both hearing the obnoxious sounds and also being scratched on their arms, because it's the one that actually gives us the stronger result, and I would not have time to go over all of them. But this is, again, the comparison where you see where the children with autism show increased activity. And what I'm going to highlight for you again, this region, the amygdala, which is important for detecting, aversive stimuli in the environment and for the emotional responses. But also there is activity here in somatosensory cortices as well in visual, in auditory areas. Okay, so this is a nice replication, and for those of you who are familiar with the neuroimaging literature, that is already a major plus, because sometimes results do not always replicate across studies. But as I mentioned, she wanted to go further, and one of the things that she did was, again, another characterization of the relationship between SOR rate and responses in a variety of brain regions. So here, we entered the composite score, and we saw where it modulated activity in the brain. Again, this is the amygdala, so areas that involving attention. Here we have the thalamus. The thalamus is a relay station in the brain. It's basically gating sensory information arriving, going onto the cortex, and also in auditory cortices and in somatosensory cortices. So far we have a nice, good replication, but really in the study what Shula wanted to do was to was to tease apart different possible mechanisms that can contribute to this effect. So do children that show high SOR, do they have a heightened response, or they may also fail to have some top-down inhibitory modulation? Do they fail to habituate, which we all do when presented with this, the same stimulation over and over again? So what she did, she broke down the responses in between different blocks in the duration of the scan. So this is, you can think about, this is the beginning of the MRI scan and this is the end. So for the auditory cortex, you see that all three groups of kids with ASD, without SOR, with SOR, and the typically developing control, they all show a sharp decrease from the first block when they were hearing those environmental sounds, and then they stay relatively low. There wasn't any significant difference actually in those groups for this condition. But for the tactile condition where they were being stroked on their arms, you see that it's only the children with SOR who do not show the sharp drop that the other two groups show. So basically, both the children with ASD that do not have SOR and the typically developing control, again, show this sharp decline, meaning, oh, okay, so this has happened before. My body is no longer responding as highly because I'm getting used to this. But the children with autism that have high SOR do not have the similar drop, and there is this significant difference. And if you're looking at the amygdala again, that basic center in the brain that codes emotional valence of stimuli, you see again a failure to come down to the level seen in the other two groups. So this lends support to the fact that one of the issues, and not so much that the difference that we observed, they were not indicative of a much higher level of sensory responses in the brain, but rather to a failure of habituation. Those regions continue to fire and send signal, this is happening, instead of saying, okay, this is not as novel anymore, it doesn't bother me much. But as I mentioned before, some other processes may be involved, and so what she did in another set of analysis, she looked at connectivity between the amygdala region and basically any other regions in the brain. So here we're looking at coupling of activity between regions that varies as a function of the task. And this was again for the joint condition where they're being stroked and they were hearing those loud noises. And there was this significant differences where the typically developing children had greater coupling between the amygdala and these prefrontal regions, which for the most part tend to inhibit, quiet down the subcortical regions. But then she teased it apart even further, again, by breaking the ASD group in two, the one that do show a symptom of SOR and the one that do not. And this is when it becomes very interestingly, because it wasn't so much the case that the typically developing kids were able to inhibit the amygdala. Those stimuli were not very aversive to them. They were habituating relatively easily. So there was some coupling, but it wasn't going in that direction. What she found is that we actually had in the group of children that do not show SOR, this very strong negative coupling, meaning that the stronger the activity in this frontal region, the lower the activity in the amygdala, suggesting that they were actually able perhaps not to have to show the symptoms of SOR because they're able to top-down inhibit some of the regions that code the negative valence of the stimuli. Hope it makes sense. Okay, so again, Shua is never stopping, and she's always thinking about the next question. And as some of you might have heard, as Dan alluded, that autism is being regarded as a disorder of connectivity. And there have been a number of different networks that have been investigated where significant alteration have been found. And I'm going to be talking to you about one that it's called the Salience network. And here you see a schematics of these regions. And these blobs are not showing brain activity, but rather they identify that highlight a set of brain regions that tend to co-activate, to work together. And this particular network is thought to be particularly important in detecting and integrating external sensory information with internal emotional and bodily states. So it's basically thought to figure out what is the most salient information that I must attend to. It could be some internal states when a baby's hungry, or when they're wet, then they need a diaper change, to something that is happening out there in the environment, okay? So it's supposed to play this role in basically allocating attentional resources either to stimuli out there in the environment or to what is happening in your own body. And it's supposed to be doing so in part by coordinating the activity of two other networks in the brain. One is the so-called default mode network, which is a network that is important in self-referential thinking and social cognition, and also in the so-called central executive network, which is a network that is instead geared toward action, planning of action in response to what's happening in your environment. Okay, so why do I want to talk about this network? As I mentioned, there has been now a number of studies that have found that it is the connectivity in this network is altered in autism, and this is a study done by somebody who was at UCLA, Lucina Udding, and in this study she looked at a bunch of different networks in autism and found that there was overconnectivity in 6 out of 10, but the one that I'm highlighting here in the top is the Salience network, and the reason why I highlighted it is because they did some further analysis that showed that this network, and this is, we don't need to go into the detail, they use a machine learning algorithm where they train basically to differentiate. They presented a lot of cases of typically developing kids with autism, look at their pattern of brain activity to then be able to predict when you feed a next set of brain data, whether that particular brain data pattern of connectivity should belong to one group or another. And what they found was that the Salience network was actually the network whereby they achieved the highest accuracy in classifying or in distinguishing basically children who did have a diagnosis for the typically developing children. And that is why, there is this hope that this Salience network connectivity could actually function as a biomarker. Okay. So, what is the next step in the work that we have been conducting at UCLA after the study that was published last year? This is really hot from the press because it just literally got accepted in the past couple of days. Shula went about addressing this issue. So far, she looked at sensory over-responsivity and connectivity in response to the task. But what about connectivity within the Salience network? As I said, this is a network that it's important in modulating attention to sensory stimuli that are internal and external. So is there a relationship here? And so again, now you see more blobs on brains, but I want to tell you that, again, these are not patterns of activation. So this is not in response to the stimuli. And during this particular scan, the children were told to simply rest and stare at a fixation cross. But we collect brain activity data, and we looked for regions that co-activate to go together during the time of the scans. And we started out with a seed in the hub of the Salience network. So we zero in this anterior insular region, which would be somewhere there, and we said, okay, show us all of the regions that are co-activating, connected, to this region of the Salience network. But the important thing, she wanted to see where this connectivity was modulated by the SOR composite scores that we had collected on the same children. So basically, we're trying to figure out to what extent the connectivity in the Salience network is modulated by SOR severity. And this is where we find that there was this increased connectivity within the Salience network hub, and again, the amygdala, the auditory cortices, and also the somatosensory cortices. So this is good because, so far there have actually been no studies that looked at SOR and functional connectivity, but most importantly, there had been no study that directly related or connectivity in this so-called intrinsic connectivity network. Again, network that are there even when you're not performing a task, and connectivity and activity during the a function that a task that you are presenting the kids with. And so this is just what I told you, that the higher level of SOR and ASD were correlated with, associated with greater connectivity between the Salience network and this region implicated in both sensory processing here and attention. But in the next step here, she, again, this is a little bit dense. I'm going to try to make it a little bit more intuitive. Here she correlated the pattern of connectivity in between the anterior insula, which is the hub of the Salience network, and the amygdala during the resting state scan when they were doing nothing, and the amygdala responses during the task where they were actually being stroked on their arms and they heard those obnoxious loud noises, because we had the same data collected in the same group of children. And there again, you have a very nice correlation. And she did the same thing for another key region, which is the region in the somatosensory cortex, where we again found this increased, stronger connectivity as a function of SR. And well, the same regions that show this increased, stronger connectivity in the Salience network when the kids were doing nothing were also the regions that showed the greatest responses when they were being stroked. So if not anything, for those of you who know about imaging, this is like quite novel to have it in together, but it's highlighting really how the Salience network can be used as a probe of sensory over-responsivity in a way. And this is good news, because the Salience network can actually be identified even in young children, infants actually. So for those of you who have heard some of the presentation in year priors, you may recall that indeed we can, and we're doing it over here at UCLA, collect fMRI data, MRI data in infants. This is done during natural sleep, so there is no sedation, nothing that could be posing any concern for the parents or for the child. And we obviously have a limited range of things that we can do in terms of presentation of stimuli because these babies are asleep. But resting state, when you just look at the pattern of connectivity in the brain, is one thing that we can do very easily, because even if there are some small differences, studies in adults have shown that you get similar resting state network, whether a person is awake and is being told to rest, whether that person is asleep, and in some cases even when a person may be in a coma. So we know that we can get at the functional connectivity in the brain of an infant when they're not asleep. Now, I don't have pictures showing all of the networks. I mean, but this is one in neonates that shows some of the hubs of this Salience network. We also get beautiful maps for the somatosensory network that shows all of the region that respond to sensory stimulation, for auditory network showing all of your auditory language areas, and for the visual system, and so on and so forth. Of course, there are some higher order networks that tend to develop a little bit later where you see the greatest changes, but this is one that you would believe should be there kind of from the get-go, because infants do have bodily sensation, and they are exposed to a barrage of new information. So it's like one that we are very fond of, and one with which we can start to look at abnormal pattern of sensory over-responsivity way before children can actually demonstrate to parents or to us that they are responding quite negatively to some of the stimuli in their environment. Okay, so this is the work. Now I'm switching gears and talk about the work that we have been doing in infants at high and low risk for ASD. Some of you may be very well aware of this approach, affectionately called the infant SIBS approach. This has been pioneered by Dr. Marion Sigmund, who was the leader of a leader in the autism community and our pillars here at UCLA. And basically the notion is, we know that the recurrence rate in families that have already a child with autism, it's much, much higher than in the normal population. And so and the rates are as high as 45% for boys that are born in a family where there is already a child with autism, which is pretty staggering. So we have some of these multiplex families in our studies, and they are very precious. But even for overall, I mean, the rate is 20%, again, much higher. So what we can do by following prospectively these infants until the age where they can then get a diagnosis, and by comparing their behavior and development earlier on, the hope is that we may be able to identify some early markers that can predict subsequent diagnosis well in advance of the full manifestation of the symptoms. And we actually have a project that is still ongoing, and one of the nice things about having been at UCLA and having such wonderful colleagues that are doing intervention work is that if at the end of our research study around 12 months of age, if we see any flags, these infants can be fed into a project that is headed by Dr. Connie Kazari, who will be telling you more about her work later on, so that they can actually start receiving some intervention that could ameliorate some of these symptoms. Now, of course, at that point, we cannot tell whether those children will develop autism or some other problems, but the notion is that it certainly cannot help to intervene to try and again steer development onto a more normative trajectory. Okay, so this, I think, should be pretty straightforward, that early identification is directly related to early intervention that can ameliorate, or the ultimate goal, even prevent the full-blown onset of the symptomatology. Okay, so after that long introduction, as I mentioned, we put these kids in the scanner these babies in the scanner when they're asleep at 6 weeks of age, and then again at 9 months of age. And while they're asleep, as long as they're asleep, we're going to get some beautiful pictures of how their brain is growing and developing. So we're looking at their structure, but that's not what I'm showing you here. We're looking at their resting state, basically network, so this functional network that I've been talking to you till now. We're also actually presenting them with language. You can do that through the headphones that we put around their ears that also shields from the noise of the scanner, because studies have shown that your brain cannot not process language even when you are asleep. So while we cannot present them with visual stimuli when their eyes are closed, certainly we can actually map this language network, but I will not be telling you about those findings today, but they're very nice. Here I'm going to be focusing on the work that Tawny has done, focusing on this Salience network. Okay, so if you look at the pictures here, these are the babies, the data from the babies at 6 weeks. This is the group of infants that are at a high risk for developing autism by virtue of having one or more siblings that have a diagnosis. And this is a control group of low-risk infants. And they're well matched by age, gestational age, and motion during scanning, because even if they're asleep, sometimes babies wiggle a little. So the picture seems relatively similar, right? So you see this is the anterior insula, which was our seed. We're basically starting from this anatomical region in both groups and say, show us where our brain regions that go up and down in this random fluctuations. And we see some clear similarity, but I want to pay attention to the fact that there seem to be some differences here in the frontal regions. And also there seems to be some differences here in what you may not infer from the slide is actually somatosensory cortices. But now, when you're just looking at the pattern of connectivity in each group, you cannot quite tell whether this differences that we see is significant or not. thought it could just be that they also show connectivity, but it's just below our statistical threshold. So what we do, we actually directly compare the pattern of connectivity that we observed in the two groups. And lo and behold, we actually did find significant group differences, which was actually almost surprising to me, because this group of high risk is composed of children that may have a diagnosis, but in a sample size of the one we're talking about here, there may be maybe 1 or 2 kids. So yes, there is such thing as the autism, the broader autism phenotype, but still the differences were more than I expected, and in particular, they were beautiful, if you will, in the sense that they made sense. So what you see here is that the group at the high risk for autism, in direct comparison to the low risk group, show stronger connectivity between that hub of the Salience network and somatosensory regions. In contrast, the group low risk for autism show greater connectivity with frontal region, including both medial prefrontal cortices and some other more lateral cortices. And why is that important? Well, remember we said the Salience network is supposed to modulate attention between something that is happening to you and versus something that is happening out there. And so this increased connectivity of the Salience network with the sensory areas suggests that those stimuli, those sensations, those bodily sensations are more salient, that drive more attention in the infants who are at high risk for autism than they do in the other group. Whereas the group that is at low risk activates a network of regions which are part of some of those networks that are more focused on the outside. Some of these regions actually are so-called in the older literature, when you're talking about older individuals, there are theories of mind region. There are parts of the brain that you use when you're trying to make sense of what others are doing around you. Okay, and there's also part of the brain here, most posterior, that it's part of that default mode network, which is, so again, involved in social cognition. So we thought these were beautiful data, which are going to be soon submitted. And I see that the time is running down, and I want to make sure to allow some time for questions, so I'm going to kind of breeze through. But I want to highlight why I think this is important. And one of the things that I have not mentioned so far is that we all know, right, that there are deficit, if you will, in social attention. So whereas typically developing babies from birth responds preferentially to both the human voice and particularly the mothers, and they also prefer to look at faces as compared to other objects. This kind of preferential or attentional biases are really not seen in children who have autism or who will develop autism later on. So in a way, this lack of to social attention remains something that the field as a whole has not been able to really to address. Some of us have done work on the reward circuitry, thinking, okay, interacting with other people is not as rewarding, that's why they may not be paying attention to that. But in essence, you still find yourself having to answer, why is it not rewarding? So of course now this decreased attentional biases, if you go by the tenets of the social motivation hypothesis, could lead to a cascade of negative consequences for later social development, because if you're not paying attention to your conspecific, I mean, there isn't much social learning that it's going to happen. But the real important question, the next frontier, at least for us and for some of the students in my lab, is to really look at whether early sensory over-responsivity may be related to this decreased social attention. Is it the case that this over-responsivity to some of the sensory internal proprioceptive feedback is actually diverting attention that in a neurotypical brain is directed toward what is happening around you, that is to what mom and dad and your siblings, et cetera, are doing? So this is some of the work that we're focusing on, and Tony is going to be relating the data that I showed you collected in the infants while they're being scanned in the MRI to other measures that we're collecting on the same infants in the lab of Dr. Shafali Jeste, who is doing both EEG as well as eye tracking. So we'll be able to see how they're paying, what they're paying attention, and relate measures to one another to begin to tackle this question. And in terms of future directions, I mentioned that there are issues with the measure that the field currently has available to assess sensory over-responsivity, but there are some strides to come up with better ways, even when just by looking at behavior, at least not to rely only and exclusively on parental reports. And of course, some of the work that Shula will be doing as part of the SFARI, et cetera, it's to look at also other physiological responses. And here, I want to highlight one more thing, since I have mentioned, obviously, that SOR is seen in other clinical populations, that one of the important things that the field could also start doing is to look at SR across different clinical populations. And this is one study that just looked at sensory evoked responses in both individuals with schizophrenia and autism, and they found that aside from the surface similarity with sensory issues, the patterns the brain informs, it gives us information that we could not derive by behavior alone, showing very distinct pattern in the two populations. So I think that is important because, again, it's only by identifying what the mechanism may be that are giving rise to the sensory abnormalities and disturbances that you can then develop appropriate and effective interventions. And with that, I'll end here. I'll take some questions, but please, if you know of anybody who has a baby, either in a family where there is autism or even in a family where there is no child with autism, please have us call us. And of course, we're also conducting a number of studies in the older children, age ranges from 8 to 16. So please spread the word, because we couldn't do it without these children and their families, which we really appreciate. Thank you.